Thank you, Yi. So this morning, we are going to have four exciting keynote lectures. Our first speaker will be delivered by Dr. Miguel Nicolelis. Dr. Miguel Nicolelis is a distinguished professor emeritus at Duke University, the founder of Duke Center for Neuroengineering. He is also founder and the scientific director of the Almond and Lady Safra International Institute for Neuroscience of NATO, and the Walk Again Project, an international consortium of scientists and engineers who are dedicated to the development of an exoskeleton device to assist severely paralyzed patients in regaining full body mobility. Dr. Nicolelis has dedicated his career to investigating how the brains of freely behaving animals encode sensory and motor information. He was the first to propose and demonstrate that animals and human subjects can utilize their electrical brain activity to directly control neuroprosthetic devices via brain-machine interfaces. So Dr. Nicolelis' pioneer brain-machine interface studies have offered potentially new therapies for patients suffering from several levels of paralysis, Parkinson's diseases, and epilepsy. Today, numerous neuroscience laboratories in the US, Europe, Asia, and Latin America have incorporated Dr. Nicolelis' experimental paradigm to study a variety of membrane and neuronal systems. His research has influenced basic and applied research in computer science, robotics, and biomedical engineering. So, Miguel, are you ready?
Yes. Thank you. I need to share my screen for a second. Let's see. Can you see the slides?
Yes.
Oh, okay. I'm ready, whenever.
All right, go ahead. Thank you.
Thank you. Well, first of all, thank you very much, Mingmin, for such a wonderful and kind invitation and introduction. Is a great pleasure, a great honor to be one of the speakers on the CIBR meeting. So I hope I can, in these next 30 minutes or so, give you just an overview of this very exciting area of research that basically comes from the interest of understanding how large populations of neurons, as you see in the left side of this slide, the first time we recorded 100 neurons simultaneously in a behaving monkey. As I'm going to show you, contrary to what most people believe, the introduction of this approach, brain-machine interfaces, came from John Chapin and I working together with an interest of creating a new approach to study how populations of neurons interact in behaving animals. In fact, that's the summary here that I'd like to make the introduction to all of you. Basically, since I left Brazil to go to the US in '89 and met John Chapin there, first as a postdoc and then as a colleague, our interest was primarily to understand how large populations of neurons in behaving animals interact as these animals perform regular ethologically meaningful behaviors, but also how these circuits underwent plastic adaptations during a variety of tasks. And by developing a new method together that we call chronic multi-site many electro recordings, we basically were able to start recording, large populations, hundreds of neurons simultaneously. And after close to 2 decades of this work, we came up across a series of principles that we call principles of neuroensemble physiology that motivated us to propose a different view on how cortical networks operate. It was to go in depth into these principles and to study further the dynamics of large frontoparietal circuits in primates that John and I came across this idea of creating a new paradigm that we name brain-machine interfaces, the possibility of linking brains directly to devices, mechanical, electronic, or virtual, was driven primarily by our basic science interest in how populations of neurons encode information. So it came to our surprise that as we implemented this paradigm in many species and then in human beings in the early 2000s, that we came up the possibility of using this paradigm to derive a series of new potential neuro rehab therapies that we have been applying since then to a variety of neurological disorders. But also, as I'm going to show, because the emphasis of the talk is going to be on basic science, this initial paradigm allowed us to create multiple other approaches, brain-to-brain interfaces and a BrainNet that I'm going to show tonight as an introduction to a variety of new fields that derived from the original description of brain-machine interfaces. Well, the classical image of a brain-machine interface is represented by this slide that John and I published originally, in which you have a brain and you have ways to sample information from this brain. It can be intracortical, more invasive methods like we use in our animals. These are arrays of microelectrodes I'm going to show you in a moment, and that allow us to record the extracellular electrical signals of these populations and in real time use a series of computational and mathematical models in the original description to extract motor parameters that these frontal parietal circuits are generating in order to move an arm or a leg in a behavior test. But the idea was to see if we could actually extract these signals and reroute them to artificial devices that could be mechanical, like a robotic limb, or electronic, or even a virtual body that this brain could actually control directly just by producing the types of a large-scale neuroensemble activity that is involved in the control of movements. So this is the original description, and of course, it was very important to realize since the beginning that it was important to close this control loop by providing both visual and tactile feedback to the brain of the subject, so the subject could have a feeling for what was being accomplished as this brain-machine interface was operating. So over the last 30 years, we have developed multiple generations of probes to sample larger and larger populations of neurons simultaneously by recording the extracellular action potentials from these electrodes. So these are two generations, the second, the third. The third we call a recording cube because we have not only recording elements in the X and Y axis, but also on the Z axis. These are microfilaments that are very flexible. They are made of stainless steel or tungsten metal filaments that can be covered with isolating material. And once implanted in the brain, they're not rigid. They can move with the brain, which gives them a very great deal of longevity. And that has been very important for all our studies because we could record, not necessarily from the same single neurons, but we could record large samples of neurons for years in each one of our animals, particularly rhesus monkeys. One of the recent breakthroughs in this paradigm was the addition to wireless technology, which allowed us, as you're going to see in a moment, that the animals could actually be recorded without any tethering to the equipment. They could be literally freely behaving and interacting with other members of their pack. So this was also a technological development that made a big difference for all sorts of new paradigms, new studies that we could not perform without the introduction of wireless multi-channel recordings. Well, this is just to give a view, a bird's eye view of one of these large-scale implants in the rhesus monkey brain. These are implants on the frontal and parietal lobes in different motor areas, primary motor areas, supplementary motor area, premotor areas, and also in posterior parietal cortex, where we can sample from the primary somatosensory cortex and areas in the posterior area of the parietal lobe. This was one of our largest samples, about 1,874 neurons recorded simultaneously from the same subject, which allowed us to create a variety of new studies in the same group of animals that we follow, as I said, for many years. So let me show you the original experimental design and how it evolved over time, so you can see how this field is being developed and how new paradigms are being added to the original classical description. This is how we started with rhesus monkeys performing very simple behavior tasks, motor tasks. For instance, using a joystick to control the movements of a cursor that has to be moved to intercept a target that appears randomly on the screen, so that every time the cursor crosses the target, the animal gets a juice reward. We did that. We trained these animals, and at that time, we were able to record about 100 neurons simultaneously from multiple frontal, parietal, cortical areas. And the extracellular activity, the combined population activity of these neurons were, as the animal was playing the game, rerouted to a series of computers that were running up to 20 mathematical models whose job was to extract motor parameters that we use to control and to reproduce the arm movements of these monkeys into a robotic arm, a 7-degree of freedom industrial robotic device that was placed in a different room and basically allowed these monkeys, as you're going to see in a moment, to implement or to utilize the brain-machine interface to actually achieve the control of this cursor when we remove the joystick. And exactly what I'm going to show you from the original video taping of these experiments. So here, you're going to see first the animal playing the game of the joystick, but in a moment later, you're going to see the first time in which we remove the joystick and we allow the animal to use for the first time the brain-machine interface and the robotic arm to control the movements of this target or this cursor to cross the target and receive a juice reward. So this is our first monkey, Aurora, who did this task. And as you can see, she's using a joystick first to acquire the target by moving the cursor. And the target is showing up randomly in different locations on the screen. And she can basically go, and when she crosses that target, she gets a juice reward. And as you can see, she became very proficient in performing this task. She could get above 95% of the correct trials. And once we noticed that, at that time, you can see lots of cables coming out because we didn't have any wireless technology. So we are sending these signals to a wall of amplifiers and computers that were actually transforming and extracting from this raw electrical brain activity the kind of motor commands that could be used to control a robotic device. And that's exactly what happened. Here, a few minutes later, we removed the joystick, so the animal could not actually control the cursor anymore by moving its own body. And we turned on the brain-machine interface so that the animal could actually now control these movements by basically using its own brain activity to control the movements of the robotic hand that was in a different room in our laboratory. And what you see here is that the animal now is using the brain-machine interface to reproduce its motor intention to actually get the robotic arm to move the cursor and cross the target so that she can get the same juice reward that she was getting when she used her own her own arm to make these movements. And see that in the beginning is a little more difficult because this is the first time that she got it right. But after a few weeks, actually 2 weeks, she was performing at the same level that she was doing when she used her own arm to control the joystick. And that was the moment we realized that this could work. Well, as time went by, we were able to show that we could get this thing a little more complex. We could expand this paradigm not only to allow the brain activity to control, in this case, a virtual arm to explore virtual objects that when the animal look at them, they look virtually identical. They are pretty much identical, but they carry a microtexture that can only be detected when the animal scans the surface of each one of these objects. There's one frequency that the animal has to identify, one surface that is the correct one for the animal to select. And it can only do that because we realized that we could get feedback, electrical feedback through microstimulation delivered directly to the primary somatosensory cortex of the animal. And that's what the reason we call this a brain-machine-brain interface. And by doing that, we demonstrated that we could expand this concept by allowing the animal not only to move an actuator, artificial actuator like a virtual arm to cross the surface of these targets, but also to discriminate the texture in a tactile discrimination task using the feedback information delivered directly to its somatosensory cortex, as shown here in B. So let me show you how that happens. So the sound in the background is of a cortical neuron. The animal is scanning the three surfaces using a typical brain-machine interface. And when it feels the correct texture of one of these surfaces that is indicated only to you, the animal didn't know that, it gets that, it selects that target, and basically gets reward for that. And the correct target is changing position in space, so the animal has to really rely on what is experiencing as tactile feedback delivered directly to the primary sensory cortex to actually get the correct texture and then get a reward. So at that point, we have just published a paper recently in 2019 expanding this paradigm to a more complex task, showing that the brain-machine brain interface really operates very nicely, and in a couple of days, the animal can perform the tactile discrimination task as if it were using its own fingertips to do this tactile discrimination task. Well, once that was clear, that was the time around 2014 that we introduced the multi-channel, large-scale multi-channel wireless technology that allow us to broadcast a large number of activity from multiple neurons, about 300 neurons, to up to 5 to 10 meters from the animal, so that we could basically liberate the animal from any tethering and allow these monkeys to perform tasks in which they were free to move around our lab. So this is one of these tasks in which the animal was trained to control or to drive electronic wheelchair using the brain activity coming from motor areas in the frontal lobe and also parietal areas in the parietal lobe combined that allowed us to test whether these animals could learn to imagine trajectories that would allow them to drive these wheelchairs to locations in the lab where they could collect a reward. And that's exactly what I'm going to show you how these animals learn to drive this device just by imagining the trajectory. You're going to see this experiment first from the top of my camera position on the ceiling of our lab. And there are 3 original positions that we can place the animal randomly. And from here, the animal has to create a trajectory to go through all the way to a target zone where we drop some grapes that work as a reward for this animal. So let's show you this first from the top. So this is about 300 neurons being used by the animal to drive this wheelchair and collect grapes in the target zone. Then we place the animal in a new area, a new position, and it has to create a de novo, a new trajectory, so that it can keep collecting grapes at libido. So the animals learn to do that. This is a frontal camera, though, so you can see that the animal gets very used to the paradigm, and he only really moves when he's getting close to the collecting zone, so to get its reward. So we learned by multiple experiments with brain-machine interfaces that you can condition these animals to stop moving their bodies and rely only on the brain-machine interface apparatus to complete the task. Well, interesting enough, when we did these experiments and we analyzed the firing patterns of these neurons in primary motor cortex and in premotor dorsal cortical areas, we are surprised to find neurons, a large number of neurons in these regions, in the cortical regions that I just mentioned, exhibiting features or tuning that is very similar to the typical place cells that we find in the hippocampus. And that was very surprising to us. We didn't expect to find that, but you can see here, this is a X and Y axis of the room, position of the chair and the monkey in the room as the monkey is moving. And in the Z-axis in yellow, we are showing the firing magnitude of individual neurons. We are showing 2 neurons here just to illustrate that beautiful place tuning in these neurons in different locations of the room. And when we combine linearly this, as people have done that for many decades in the hippocampus, we could reproduce the trajectories that our monkeys had to perform these tasks. So we found in very lower levels of the so-called cortical hierarchy, a very complex activity. And this was not only the case. I don't have time to show all the interesting parameters that we derive from these animals, but we also we found cells, about 30% of the neurons that would encode the magnitude of the reward that the animals were receiving in tasks like that. So neurons in the motor cortex were multitasking in a very interesting way, in a very complex way, not only coding for motor parameters, but all sorts of higher order parameters. Well, another new area that came about with this advancing our ability to record wirelessly from brains was the possibility of creating shared brain-machine interfaces, or as I like to call, brain nets. In this case, more than one animal can collaborate because we can combine the brain activity of more than one subject to produce an output, a complex output that can be used to control a particular virtual actuator, like a virtual arm, like I showed you before. The easiest example is represented here, where monkey 1 is responsible for encoding the X dimension of this virtual arm movement in space. It's the same task that you saw before with Aurora, while monkey 2 is responsible for encoding the Y-axis. By simply summing the activity of these two brains, without the animals even knowing that they're collaborating, we're able to provide or to generate these two-dimensional outputs that allow the animals to share a brain-machine interface and also share a reward that they receive it for doing this task. But we generalize this task for more than two animals. Here, you're going to see the outputs, the two-dimensional outputs of three monkeys that don't know that they're collaborating, but monkey red here is providing control for the XY dimension of the movement of this black spot. The green monkey is providing Y and Z, and the blue monkey is providing output for X and Z. And by summing the three brains, we generate a three-dimensional output that controls the movements of this black circle that has to be placed continuously inside this circle for the three animals to share a reward. And even though they didn't know that they were collaborating, we're able to get them to basically perform these brain net tasks very well and coordinate their outputs so that the black spot can be continuously driven inside the target. That was the objective of this paradigm, and we noticed that the monkeys get adapted to it very well. They relax and they combine the activity. But how do they do that? To figure out what was going on here, we decided to generate more realistic tasks, social interactions between our monkeys. And in this case, the alpha monkey of our colony is responsible for driving this electrical wheelchair, as you saw before, towards the target zone where he's going to get the reward, as you saw in the previous example, in the previous experiment. But now in the room, there is a lower ranking monkey that is observing the boss to drive around. And as they interact socially, because they establish eye contact, you're going to see for a moment that this monkey gets closer to the lower ranking monkey before it drives towards the target. We examined what was going on in both brains simultaneously. And as you can see here, the first monkey is driving the wheelchair using a typical brain-machine interface, as you saw in the example before, and now is driving towards the target. It takes a little moment to look at the lower-ranking monkey, and suddenly it changes its attention to go and collect the grapes. But once this guy collects the grapes, the lower-ranking monkey is going to get a free juice as a reward for observing the boss to do the main job. So we repeated this experiment many times using either our higher monkey driving or the lower monkey driving the wheelchair while the other monkey observed the task being performed. And to our surprise, when we look at the brain simultaneously, this is the observer, lower-ranking monkey, and this is the driver, high-ranking monkey. In each line, you see individual neurons recorded in the primary motor and premotor dorsal cortex of these two monkeys. Yellow bars represent extracellular action potentials. And what you see here is that during that excursion that you just observed, there is a high level interbrain synchronization that occurs. Look at this. There is action potentials being generated in both brains almost at the same time as the monkey is driving to collect its reward. So this was the first demonstration in monkeys, in non-human primates, of a high-level interbrain synchronization in animals engaged on a social task. And in the last couple of years, since this paper was published, many reports have been published in humans showing the same level of interbrain synchronization during a variety of social interactions. Interesting enough, when we look at what this interbrain synchronization coded for, we could see that we could predict where the wheelchair of the driving monkey was located in space by looking at this synchronous activity. We could predict the velocity, but not the acceleration of the wheelchair. But more important than these parameters, we could actually predict who was driving the wheelchair, the social ranking of the monkey in driving the wheelchair, because when the alpha monkey was driving towards the observer lower ranking monkey, the interbrain synchronization was the highest possible. When we inverted the paradigm and we put the lower ranking monkey to drive, there was some level of interbrain synchronization, but was much less than what we observed when the higher order or higher ranking monkey was driving the wheelchair. So we could extract social parameters from this interaction between two brains, something that has now been reproduced in humans in a variety of scenarios. So that was a very, very interesting outcome that we never expected to get from primary motor cortex, the encoding of social hierarchy or social relationships in a group of monkeys. Well, I showed you all the basic science or some of the basic science that we have done, but of course, I couldn't finish this talk by showing that since the beginning of the year 2000, John Chapin and I had proposed, actually in 2002, the brain-machine interfaces could use to benefit patients who had suffered from severe brain lesions, like spinal cord lesions that made the patients severely paralyzed. We knew that these brains continue to encode motion in a different way, but they remain for life to encoding information that cannot reach anymore the body. That's the reason patients remain paralyzed. So we propose in this article that perhaps in the future, brain-machine interfaces could be used to allow these patients to reroute their brain activity to a new body, a robotic vest that would allow these patients to actually regain mobility by using a brain-machine interface. That's exactly what we tried to do when we created the Walk Again project in 2013. And by collaborating with an international group of scientists and physicians and neuro rehab personnel, more than 100 people together, we defined our goal to see if we could actually use a brain control exoskeleton to allow patients to regain mobility and use their brain activity to basically control the movements of their lower limbs again, bypassing the spinal cord injury. For that, we selected a group initially of 8 patients that was increased to 20 patients, in which patients had been living with a chronic spinal cord injury that rendered them completely paralyzed and without sensitivity below the level of the injury, some of them for more than a decade. And then we put them into a paradigm that combined virtual reality, robotics, and brain-machine interfaces using a non-invasive method, electroencephalography, to see whether they could learn to control a custom-designed exoskeleton and regain mobility by using the idea that I have just described to you in primates. But again, this was a non-invasive method. This was one of the key inventions of this exo because it was instrumented with these printed circuit boards with pressure and distance sensors, for instance, in the plantar surface of the feet, to generate a tactile feedback every time the exo touched the ground to allow these patients to have a feeling of what was going on, because this feedback was delivered to the only part of their bodies that were secure, where we knew that they could feel. The skin surface of their forearms. Here you see one of these patients training in an immersive virtual reality environment where the brain activity is being used to control the avatar, the movements of an avatar soccer player walking on grass. And every time that this avatar touches grass, the patient receives a wave of pressure signals delivered to an array of vibrating elements to their forearm skin. And he's using this feedback to allow them to get a feeling, a tactile feeling. Now the patient is walking on sand, as if he were walking on a beach. And we realized that we could induce these patients that were paralyzed from the chest down to actually experience a phantom limb sensation, something very similar to a phantom limb sensation, because before they even went to work on the exoskeleton, they were reporting to us that they felt when they were in this immersive environment that they're walking by themselves. Well, this is where the moment in which for each one of these 8 patients, we put everything that I described briefly to you together. This is a T6, thoracic 6 level patient, paralyzed below the level, who is using now a EEG brain-machine interface to control the movements of an exoskeleton. And as you're going to see, as the exoskeleton allows the patient to walk again, is getting tactile feedback so the patient can feel the ground. There is a large mirror in front of this patient, so he's getting a visual feedback, but he's also getting tactile feedback as it is using its own electrical brain activity to walk around and get through this about 5 or 6 meters of walking in the beginning of their training. And you just need to look at the patient's face to realize how surprised and how elated he is about, for the first time in 6 years, walking again. Well, I don't have time to show you the actual kick that we delivered. One of these patients delivered in the opening ceremony of the World Cup. It was a major event here in Brazil and worldwide, but I want to finish by showing that something that we never expected to happen. After we did the demonstration, after 6 months of training, we brought these patients to the lab for a comprehensive neurological examination because that was mandatory in our clinical trial. But of course, we didn't expect to see anything because the patients had remained at the same clinical level throughout their lives after the spinal cord injury. Well, it turns out that when we brought the patient who delivered the opening kickoff of the World Cup, Juliano Pinto, he was a T4 for 10 years, meaning he had no movement and no sensation below that level. But on July of 2014, that guy was classified as a T11. He had recovered over 6 months of training about partial tactile sensation over 7 dermatomes below the level of the injury. That was a total shock for us. But we start doing this for one patient after another. This is in red, is another patient, similar result. So when we measure this over a period of 28 months since the beginning of the training, twice a week, training with the exoskeleton once a day, using the apparatus that you just saw, the average recovery of sensitivity was about 10 dermatomes for the population of 7 patients. This was something that we never, ever expected and had never been described by chronic spinal cord injury patients. Simultaneously, when we saw this tactile recovery, we tried to test for what the patients could be doing motor-wise. And to our surprise, after 13 months of training, one of our patients that was paralyzed for 14 years started showing these movements of the right leg, compound movements of the right leg, despite the fact that she was classified for more than a decade as an Asia A, a complete paraplegic at T11 level. Well, 1 year after that, we continue to follow all the patients. This is the same patient a year later. Now we are recording EMGs all over her legs. That's the reason you're going to see lots of wires coming out of her body. But she surprised us again in the same task where she was suspended, and we asked her to move. And she basically started moving her leg, but then the other leg. She had not moved the left leg when we started showing, or she started showing recovery of these movements. She was only moving the right leg for about a year. But then about 22 months into the training, she was basically able to move both legs in a compound movement, showing, again, a partial motor recovery. There was actually paralleled by a partial visceral recovery because all of our patients show a significant degree of improvement in bladder control, in bowel movement control, and sphincter controls. So they had partial tactile recovery, partial motor recovery, and also partial visual recovery that culminated after 28 months. When we look at all these patients together, they started, as I mentioned to you, 8 patients started as an American classification of spinal cord injury as Asia A or Asia B, which means complete paraplegia, no sensory feeling below the lesion and no movements. By the time we ended, one patient had to drop out of the study with 12 months. He had to move out of São Paulo, so we couldn't continue to follow him. All the 7 remaining patients after 28 months evolved to become Asia C, or partial paraplegics, something that, again, nobody had reported as this number of patients at this magnitude of partial neurological recovery in cases of spinal cord injury, complete spinal cord injury, after so long of a period. And this is just to show that a couple of these patients had such a major neurological recovery that when we gave them some body support, a little cart to move, and basically some spinal cord stimulation, they were able to actually generate, without the exoskeleton, without using a brain-machine interface, just to control the delivery of this electrical stimulation to the surface of their skin, of the legs, they were able to generate spontaneous patterns of movement. And they were able to walk with this about 60 to 70% of body control and a little bit of help from this cart and this functional electrical stimulation, non-invasive, of their legs. So I'm also glad to report that in the past 5 years, we have been able to establish a wonderful collaboration with our colleagues at the Xuanhu Hospital at the Capital Medical University. And after the reproduction of this protocol there, our colleagues are reporting very beautiful and similar results as what we saw here in our lab in Brazil. So I'm very happy that this collaboration was very fruitful. And basically, I use this case and this very quick review just to show you how a basic science idea that was created to allow us to improve our ways to measure and study how populations of neurons work in freely behaving animals eventually led to a very important advance for spinal cord neurorehabilitation. Thank you very much.
Thank you so much, Miguel, for the exciting talk. So we have received a few questions from the online audience, but
sure.
Let me rephrase them. So many people are very interested in the final part of the talk about the training of paralyzed patients. So they want to know, where do you record the signals? And how do you control? Did you deliver stimulations to the spinal cord?
No, we didn't use any electrical stimulation of the spinal cord in these patients. That was done for Parkinson's disease, a complete different protocol. Here, we only use EEG, about 16 to 32 channels of non-invasive electrical encephalography. And we basically measure the inhibition of the mu rhythm as a signal to control the intention of the movement. And that intention was delivered to our software that allowed the control of the 12 degree of freedom of this exoskeleton. These were electrical hydraulic motors that were triggered by the motor intention of the subject to walk. And we also discovered that we could make the patients alternating the left and the right hemisphere to generate a synchronous alternation of right and left leg movement. So Juliano Pinto, the guy who delivered the kickoff of the World Cup, we were able to extract 3 beats out of the modulation of his mu rhythm. He was able to actually get multiple levels of modulation of mu rhythm. And that gave us a higher level control than you usually get when you use just the EEG. So, but there was no electrical stimulation of the spinal cord in these spinal cord injury patients. We didn't need that.
Okay. So let me ask a question. Actually, that's my own question.
Sure. So,
we have witnessed substantial progress in the field of the brain-machine interface. So I'm trying to find out what are the major challenges that we need to overcome to see a revolution in the BMI.
Well, it depends. If you're talking about basic science, I think in my opinion, as I mentioned to you before, the frontier is in the brain net, in recording from multiple animals in more sophisticated social tasks, so that we can understand what is that this interbrain synchronization is capable of encoding, what kind of high cognitive and social parameters, in addition to motor parameters, we can extract from this interbrain synchronization. For instance, I just read a recent paper on a quartet of musicians playing classical music, and what happens on the interbrain synchronization, and how different areas of the brains of the different players get synchronized at different moments of the music, and how you can predict who is going to lead. So if you have a violin, two violins, and a piano, and I don't know, a flute, you actually can show by the level of interbrain synchronization who is leading in the next few hundred milliseconds the next piece of the music. So there are very interesting aspects. And in that sense, I didn't have time to show because of the time constraint. We use a brain net already between a spinal cord injury patient and a physiotherapist to combine their brain activity to make the training easier for the patients. And it actually worked. And it was the synchronization of the two brains that helped us do that. Because as you can imagine, a naive patient has a little bit of difficulty to understand what is to use a brain-machine interface, but the physical therapist was overtrained in that task. So her brain was way synchronized. So when we merged the two brains, we gained time in the training, and we made the patient understand better the concept of making this paradigm. And the good part is the physical therapist didn't need to be next to the patient. It could be in a different city or in a different location. So we could merge in a brain net fashion the activity and get the physical therapist to train multiple patients simultaneously. On the clinical side, in my opinion, it depends on what kind of disease, neurological disease, you are focusing on. You're focusing on diseases that generate very high levels of body paralysis, like lock-in syndrome. You need to improve the number of neurons that you can record simultaneously from inside the brain. So you need to get to a few thousand neurons. My goal in life is to be alive to see between 10,000 and 50,000 neurons being recorded simultaneously, and in a wireless way, in an implant that you see nothing, and is very safe, and you can now use these signals to generate multiple outputs. So some may go to the spinal cord, let's say, if you want to stimulate the spinal cord. Some may go to a robotic exoskeleton that is very light and is very easy to operate. But some may go to control other devices that the patient may need to improve his or her quality of life. So your question depends on what area you want to address, if it's more basic oriented, basic science oriented, or more clinically oriented.
Okay, so we still have a couple of minutes for a final question. There's a question from the online audience. So they're wondering whether Parkinson's disease patients have ever had rehabilitation experiments, using the brain-machine interface.
Yeah, I have published almost in 2009, we published this in animals, but then recently about 100 patients have tested our technology of a brain-machine interface that stimulates the dorsal column of the spinal cord And people look at me and say, well, that has nothing to do with Parkinson's disease at all. Well, it has nothing to do if you look at the Parkinson's disease in the classical way. A dopaminergic disease, or dopaminergic neurons die, and then you get these abnormal patterns of brain activity that make people tremor and not be able to walk. Well, I look at Parkinson's disease as producing, by the loss of these dopaminergic neurons, a type of focal epileptic kind of activity, hypersynchronization. And what we discover in humans and in animals is that if we stimulate a high frequency on the dorsal column of the spinal cord, we can basically disorganize the seizure attack. The epileptic activity goes away, and instantaneously, the animals or the patients can move, can walk, without even taking L-DOPA anymore. So this has been tested, as I said, by many small clinical studies in Canada, in Brazil, in Europe. Now it's starting in China. And we have about 100 patients around the world. The largest sample is in Canada and in France, where people have used this technology in terminal Parkinsonian patients that don't respond to DBS, don't respond to L-DOPA anymore, and they were able to move, to regain mobility, and these are freezing patients, we call, patients who suffer from freezing gait. And they were able to recover to a level that surprised a lot of people. So I'm putting a lot of effort on testing this new therapy too, lately.
Okay, great. I think time's up. We are still receiving many questions. We'll leave this question for private interaction discussion. So thank you again, Miguel, for a very exciting talk. I look forward to chatting with you later. Yeah. Have a good night.
Thank you, Mimin, for the invitation. Have a great meeting.
Yeah. Great. Thank you.
Bye-bye.
Bye.